All right, welcome to Module 3. This is a whole week on stems, leaves, and water. You're going to see a little bit of repetition from the following 2 weeks, which I hope is helpful for you, as well as some new topics as well. All right, so in your book, you're going to read about stem, leaf, and leaf tissue. So that's where you'll see some review. We'll look again at a cross-section of a leaf and just some review on the structures there. This cross-section of a stem and review on some of the structures there. And then we'll look a little more closely at leaves as well and some of those structures. We've seen the epidermis in the context of trichomes and some of those basic tissues, and we'll get a little more into some of the internal structures of the leaf. We'll also take a look at another structure on the epidermis, these stomates that are really important in water movement throughout the plant. And so you'll get into a little bit of how that all works as well. All right, so for supplementary animations this week, there is an overview of transport in the plant, again, talking about xylem and phloem. Some basic videos on diffusion and osmosis, which are kind of key to understanding these movements. And again, we're going to keep this at kind of a high level. We won't get into the nitty-gritty details, but I do want you to be comfortable with these ideas. And then here's a video on xylem and phloem transport in particular, just kind of getting a little more into the specifics of that in the context of diffusion and osmosis. All right, and so on the cannabis end, we're going to be looking at stems and leaves, and we're going to focus a little more on leaves this week. So we have kind of looked at stems a little bit last week as well. But again, we're kind of looking at those structures of the stem. This is another visual that might be helpful for you. Again, showing the bast fibers on that outer cortex along with the phloem, and then the inner core containing the xylem and the pith, the lignous pith. There's also here for you some of the cannabis leaf and stem anatomy that you can use while you're reading in your book. It could be helpful to have this also open so that you can compare the two, and you're going to see a lot of commonalities between what you see in cannabis and other dicots. I'm not going to point out a lot of specifics here, but I guess except for one thing here, I just want you to look at these two middle panels, which is the cross-section of a leaf. And I want to point out these trichomes again, but just to note that on the underside of the leaf, there are these they're non-exuding, so these are trichomes that aren't producing volatile molecules down here. And then up on the upper surface, there's some of these smaller trichomes, some of which do produce secondary compounds. There's a lot of variation here, so if we just look over on this side, there's a couple glandular trichomes here on the underneath side, but here you just kind of get a little more close-up view of that leaf, and you'll just see there's a lot of surface features to it that give it a lot of interesting chemistry in particular. All right, and then another thing I want to point out to you is just the basic anatomy of the leaves and the flowers, which we'll get into the flowers a little more. Let's see, I'm just looking here more in week 5. So anyways, I just want to give you a little overview to the overall morphology of the above ground tissue. So we have our stems here, And this paper, everything I'm talking about is in this paper. So if you want to open up the slides and look at this link, you can get into this in more detail. It might be really useful for you to do so. Okay, so what we have is the main shoot here with an offshoot called an axillary shoot coming off. And then at that point, a leaf that's emerging. And then kind of around there is some of the structures I want to point out. So coming off of this junction of the what's called the foliage leaf or the true leaf and the main stem is a bract. And so this is a modified leaf. It's going to kind of support, it's going to be an indicator, a support, just kind of right under the flower. So on each side here we have a bract and then a flower. And if we look at that flower in more detail, we can see one here. So we've got an axillary shoot and a bract on each side, and then the flower. The flower itself consists of two other structures. The first structure is marked with an R here, and that is the perigonal bract, which is indicated by these circles here. So this is the perigonal bract, and then coming out of that are the stigma, which are the structures that are going to accept the pollen, and then wrapped by this perigonal bract is the ovary. So I'm kind of going into a lot of detail here. We're going to bring it up again in week 5, but I just want you to be able to piece out the true leaf and this modified leaf, the bract, and then the flower, which contains another bract. And the reason this is all important is because these bracts and perigonal bracts, as well as the leaves, are all producing high amounts of cannabinoids through the trichomes that exist on those structures. So that will be important in your reading for this week. And again, it comes back to these trichomes, which I've shown you in the previous couple weeks. Some new additions. You can see a stomate here in context with some of these more glandular trichomes that are doing more of that cannabinoid production. So that's just a scale view there. But you're going to really get into this in your reading, so I won't go into more detail here. All right, so the first paper that you're going to read for this week comes from a group in Luxembourg, which is a small country about the size of Rhode Island in between France and Germany. And they've written, this group has written this review on the biomolecules produced by cannabis. And you'll recognize some of the images. So I've taken some of the figures that I've shown you in this slide and previous weeks from this paper. So we're going to finally get into it, and I think you'll enjoy it. The second paper comes from a group in Estonia. So this is a country in Eastern Europe that borders Russia. And this paper is about, it's a review article now about the use of cannabis for biofuel production. And we'll get a little more into the cellulose versus lignin available, depending if we're thinking about that cortex versus the core of the stem. All right, so your optional extra credit assignment this week is going to be to identify the true leaves in your bean seedlings from the cotyledon. So you can use figure 828 of your book along with the assignment posted to do that if you would like. And as usual, I hope you have a great week. I hope you are reaching out, and if you're not, please do so this week with any questions, comments, anything, just to touch base. And a reminder to come see me at office hours Monday, Wednesday, and Friday. All right, I hope you have a great week, and I look forward to working with you on this module.